there's been a big explosion in the knowledge of genomic and genetics for pulmonary fibrosis. As the conference explained, the variants that are linked to the disease can be basically broken down into common variants. They're very common in the population, as well as rare variants. And right now, a lot of emphasis is going into using that genetic information to really precisely tailor therapies to individual patients based on their inherited variants, both those rare and common variants. And so the conference went over basically the developments over the last 10 years in terms of the discovery work that's gone on in terms of what genes and what variants in particular have been linked to the disease, and how that information offers prognostic information for the patients in terms of predicting what their disease course may look like, as well as what medications they may respond to in a positive way or would negatively respond to. And so that's been a big change over the last 10 years in terms of what we understand about the genetics of pulmonary fibrosis. Another big development in the field has been the application of a technique which is called single-cell RNA sequencing, the application of that technique towards the disease idiopathic pulmonary fibrosis. And so the reports coming out of the meeting suggest that now we can look at how each individual cell in the lung is expressing a profile of genes and proteins, and we've never before had that type of resolution. And so the application of the technique towards IPF, now multiple independent groups are reporting that the singular cell signatures that characterize this disease. Now, we don't yet know how we can leverage that information to help patients, but it's just such a novel finding. They're reproducible findings, and so that was those data were presented at this meeting.
At a recent session of genomics and transcriptomics in interstitial lung disease, there was a very nice review of telomeres, how they function, and how genetic polymorphisms in the genes that control the enzymes that affect telomere length are associated both with the incidence of idiopathic pulmonary fibrosis as well as the outcome of idiopathic pulmonary fibrosis. And now it's known that for other non-IPF interstitial lung diseases, telomere length also correlates with outcome, so that shorter telomere length correlates strongly with a worse outcome. The relevance to this in terms of familial interstitial lung diseases is very strong, since a number of different genetic polymorphisms in this pathway have been identified. And then the question is whether there's any therapeutic potential of targeting this pathway in the future. And that will be the subject of future research. In terms of transcriptomics, there was a beautiful presentation on the use of single-cell gene expression in IPF and the new insights that that's led to, including the recognition of aberrant basaloid stem cells that are the progenitor stem cells for the airway epithelium. And these cells appear to have abnormal gene expression. And this was particularly interesting to me because in our research lab, we are also culturing basal cells from brushings of small airway epithelium. And we've also seen changes in gene expression that potentially could explain part of the pathogenesis of the fibrosis of idiopathic pulmonary fibrosis. So this is a real novel and very state-of-the-art type of research project, and very exciting for the research community to see this.